Hey guys, all right, go ahead and open up your cellular respiration notes. And go ahead, while you're doing that, I'm going to just point out that cellular respiration is the opposite of photosynthesis. So if you recall, we were talking about photosynthesis taking carbon dioxide, water, and sunlight, converting it into glucose and oxygen. Now we're going to do the inverse. We're going to take glucose and oxygen. We're going to convert that now into ATP, adenosine triphosphate, and then the byproducts of carbon dioxide and water, or H2O. All right, so let's get on into it. So your notes should line up very nicely. And first, before we can get into cellular respiration, we have to understand the difference between anaerobic and aerobic respiration. So I realize you guys can just hit pause, fill in the notes that you need. I'm just going to kind of talk through some of this for a few. All right, so to get started, cellular respiration either occurs in the presence of oxygen, meaning that there's O2 available for the process to take place, or without oxygen, or not in the presence of oxygen, and that is what we call anaerobic. So when you see an, that's the prefix for not, and then aerobic with oxygen. Now the big deal here is that aerobic, when we have oxygen available, we're going to get a high yield of ATP. We're going to get a lot of ATP possible. And best case scenario, we could be talking anywhere from 34 to 36 ATP. So that's something right off the bat I'm going to want you guys to just jot down, right? So we're talking 34 to 38 ATP under ideal conditions. When we talk about anaerobic, 2. And that's obviously a big deal because we remember that ATP is going to contribute energy for cellular work. So we want our maximum yield when we break down a glucose so that the cell has all the energy it needs to be able to do work. Now, I like this slide because it points out that we have 3 stages in aerobic cellular respiration. So you got the glycolysis, you got the Krebs cycle, and you got the electron transport. So this is going to be important as we move forward. We're going to build on these ideas. All right. Now, pull to the next slide. Glycolysis, the splitting of glucose. So this is going to be the first step in both anaerobic and aerobic respiration. The splitting of glucose is going to allow for 2 ATP to be made. And this is kind of a critical point because in an anaerobic cellular respiration situation, you have no oxygen, you're going to get a quick 2 ATP, and then the process is over. But it's just the starting point for aerobic cellular respiration. Which brings up a really good point. Down here you see that this is believed to have started before the rest of the stages for cellular respiration. And if you go back to your earth science, and for those that haven't taken earth science, I apologize, but life on earth began with cyanobacteria in the ocean. And for those that want to go back and read scripture, God created the sea creatures and then the land creatures. So the cyanobacteria begins in the ocean, and oxygen doesn't exist yet. It doesn't exist until life starts the photosynthetic process and starts to make oxygen a possibility. So the need for energy was still there, but the oxygen wasn't available. So that's where we look at glycolysis as photosynthesis was able to take place, but we were only able to get the ATP for the work that needed to be done in that cell. Later, as oxygen becomes available, larger and larger cells and then organisms are able to develop, all because of the presence of oxygen and the various steps that we're going to see in a second. So I want you guys to remember this slide. It's going to be important. We're going to build on it. This chapter moves pretty quick, but it's all building blocks on things that we've talked about. All right, so we understand we can have aerobic and we can have anaerobic cellular respiration. We understand that anaerobic is not going to have oxygen. All right, I've said it enough. Let's move forward. What exactly is cellular respiration? Right, it's the process that releases energy from glucose to make ATP. Now we saw that ATP was produced during photosynthesis in the light reaction when we had the ATP synthase that was pumping out ATP that really was only in there to help build the glucose in our simplified version. Again, don't go off to college and then come back and be like, Mr. Weavers, I learned. Yes, I know there are other things going on. We're just going to stick to what we've learned right here. So far, we build the glucose using the ATP. And as I said, in building that glucose molecule, there's a lot of energy that's been trapped in those different bonds. So if we can break those bonds, if we can utilize the energy that's built up in that glucose molecule, well, then we can unlock a whole lot of energy. And now the cell can do a lot more work. So going back to our understanding of macromolecules and how we build storage with starch, we build storage of energy and glycogen. Well, then if we can harvest that energy by breaking down the bonds of glucose and now break the actual glucose molecule into its smaller components, we can now start to be able to get more work. So this is going to involve the chemical reaction. You're going to need to write this in. C6H12O6 plus 6O2 yields 6CO2 plus 6H2O plus ATP. And we said before that that is the opposite reaction of photosynthesis. Now, this is another one of those equations that you're going to need to just memorize. You're going to need to know it front and back. And I would certainly say right now, it's trimesterable material. Both. Whether you're talking about photosynthesis, cellular respiration, it's a critical point in this first trimester that you guys understand these reactions and you understand the stages. So photosynthesis, again, a little comparison. We had the light reaction and then we had the Calvin cycle. Well, cellular respiration, aerobic cellular respiration, has 3 stages. And glycolysis is stage 1. Krebs cycle, also known as the citric acid cycle, stage 2, and then your electron transport is going to be stage 3. Now, you noticed I said you have 3 stages in aerobic cellular respiration. If you look over here, if we have anaerobic cellular respiration, well, glycolysis can happen, right? Because we don't need oxygen, but it can certainly be available. But to move on to the next stage in the Krebs cycle or into the electron transport chain, that's where we need the oxygen. So these two, right, are going to be your aerobic, and anaerobic or aerobic will have glycolysis. So you can pause for a second, you can look back at your notes, and you'll see glycolysis is noted in both. But where they take place would be important. So again, when you go through and read through your notes tonight, because you finally bought into that idea, read my notes every night, be prepared for class, get really nice grades, you got to know where these different things take place. So glycolysis is going to take place in the cytoplasm, and specifically the cytosol. So again, tying back to the last chapter, knowing that you had the light reaction in the thylakoid and then the Calvin cycle in the stroma. Here we want to look that the glycolysis takes place in the cytoplasm. And then once we've actually broken the glucose molecule into our pyruvate, which we're going to get into in a second, that then goes into the mitochondria where the Krebs cycle or citric acid cycle can take place. And we'll get into that a little bit more. The electron transport, which we mentioned electron transport when we looked at photosynthesis, and you saw in the light reaction after photolysis that energy was going to move down the chain. Well, here, the electron transport is going to actually generate a lot of ATP. So this diagram is actually really helpful to start to visualize what's going to be taking place. And I'm going to tell you right now, if you do nothing more than memorize this diagram and what happens at each stage, then you're head and shoulders above those that just go, oh, I'm gonna read, because this flow chart really breaks it down. So we start out with our C6H12O6, our glucose. Now, to get glycolysis to work, you need to input 2 ATP. And I think about that like if you ever play scratch-offs. I come from an Italian family, so your Christmas stocking always had scratch-offs. You got to buy the scratch-off to try and win some money. And by the way, I do not promote gambling, but it was a family thing. So the initial investment of 2 ATP yields 4 ATP. But really, because you already had to spend 2, you've only gained 2. And that's where you see in this diagram that there's a net of 2 ATP right over here. Right, so you could say in glycolysis, 4 ATP will be released. But really, how much have you gained? That's 2 ATP. All right, and then we see you can also have NADPH coming out. You come down from glycolysis and you have 2 pyruvate, which is taking a C6H12O6 and breaking it into 2 3-carbon molecules. So you go from a 6-carbon to 2 3-carbon molecules. Now, both aerobic and anaerobic can do this, but only aerobic can continue down into the Krebs cycle. So now we're going to go from the cytoplasm into the mitochondria. And this is where things get fun. 1, we're going to generate CO2, one of the byproducts. And 2, we're going to pick up another 2 ATP. All right, so now really we've doubled our initial investment, right? So you put in 2, you gain 2. But now you pick up another 2 from the Krebs cycle. So you put in 2, now really you're ahead by 4. Okay. Mr. Weavers, that's really not going to help much. And this is where the electron transport, or what we call phosphorylation, comes in. This is where, under ideal conditions, you can get a gain of 32 ATP, which now can bring our yield all the way up to 36 ATP. And that's where I said 34 to 38, depending on how efficient things are working. So you can very quickly see the electron transport is where the big gain is, right? And it's kind of like when somebody puts money in the stock market. Well, they make a little, okay, I'm going to take it out. Put a little, I hold a little while, I take it out. Or I put in and I ride the market, and boom, I get the big grab. So that's what I look at if you want something to reference for your electron transport chain. So for glycolysis, stage 1, cellular respiration. Again, the primitive, the initial pathway for energy does not require oxygen, so happens with aerobic or anaerobic cellular respiration. And we're going to see glucose, a 6-carbon atom, split into 2 3-carbon atoms. So what exactly is going on here? So when we start glycolysis, energy is required to get it going, right? I said you need to have that initial investment. And what's really happening is you start out with a glucose molecule, and it gets rearranged so that you get 2 phosphate groups that are going to be attached. So now you're thinking to yourself, where would I get a phosphate group from? Well, I guess that's where the ATP comes from. Remember ATP, adenosine triphosphate. So you're going to pick up that molecule, and in turn, the ATP now becomes ADP, and the glucose molecule plus the two phosphates now become unstable. So picture it, glucose was totally happy being a 6-carbon molecule. But suddenly it's got these two phosphates on the end, becomes unstable, and now it's actually set up to be able to split. Now, a little disclaimer, there's actually a 2-step splitting process. We're not going to get into the technicality on that. However, the net result is you end up forming a pyruvate, and you get 2 ATP and 1 NADH molecule for each one. So you took 6, you made 2 3-carbon atoms, and therefore you're actually picking up 4 ATP and 2 NADH. But remember, that's what you gain. It's not necessarily your net, because you had to put the 2 ATP in to get it rolling. I know I keep emphasizing this, but this tends to be the point where students struggle. How many ATP did I get from this one? How many did I have to put in? Things like that. And that's what this next set of notes really emphasizes. Follow the energy. 2 molecules of ATP to get started. You get 4 ATP, right? 2 from each pyruvate that is formed. And therefore, when you do the math, you got 4, but you had to put in 2. So really, we only have a net gain of 2. And then we said during that stage, you're going to get 2 NADH, also as a product. So remember, products, reactants, those things are important. Also remember, when we're talking about NADH, we're talking about an electron carrying or an energy carrying molecule. So just like when we talked about NADPH and NAD+, here we're just talking another molecule that can help us to move energy along. And that's what we see then in stage 3 when we go to the electron transport chain, or phosphorylation. So now you all have this little diagram, super helpful, little sketch. So by all means reference this. You see the 2 ATP going in, you can count the 4 ATP coming out, and your pyruvate formation. A little reminder that glycolysis took place in the cytoplasm, but now the pyruvate produced during glycolysis enters the matrix of the mitochondrion, where they start the Krebs cycle, or the citric acid cycle. So this is where things are going to get a little more interesting. Before it can start the process, your pyruvate is split apart using a coenzyme A, right? And that's going to form acetyl coenzyme A, carbon dioxide, and more NADH. So these are all products. Again, if you're looking at the reactants versus products, we put in pyruvate. What we get out is acetyl coenzyme A, carbon dioxide, and NADH. So now you got the Krebs cycle, right? It actually begins with that acetyl coenzyme A, and that's going to be converted into citric acid, or citrate. And during this reaction, we're going to, and by the way, there's, again, more detail that could go there, but during this reaction, you're going to have energy released, and we're going to have more electron carriers, so NADH and FADH2, plus 2 more ATP. So now we started the process with glycolysis. We picked up 2 ATP. Then you come down, you pick up 2 more ATP from the citric acid cycle. Carbon dioxide is also given off as a waste product. So that's where we're going to start to pick up the carbon dioxide. Remember, we're trying to fill out or balance out that equation. And now we're all set to go into the electron transport chain. So I'm going to stop here, and the second video that I've posted is going to give you guys, I think, a better visualization of seeing how these processes take place. So please take a look at that, and we'll pick up here tomorrow.